Good evening, and welcome to The Gentle Scientist. Tonight, we'll take a gentle walk through a hidden kingdom, one that most of us pass every day without truly seeing. Beneath our feet, in the soil, in the shade of fallen leaves, and even in the air we breathe, lives an ancient civilization, silent, soft, and alive with secrets. It is the kingdom of mushrooms, the quiet architects of life and death, and the unseen network that ties the earth together. Mushrooms are strange, aren't they? They appear overnight, sprouting in circles after rain, here one moment and gone the next. To many, they are simply odd plants, damp and fleeting, growing in places where decay has settled. But in truth, mushrooms are not plants at all. They belong to their own kingdom, the fungi, a lineage older than trees, older than flowers. The story of fungi is not the story of petals or leaves reaching for light. It is the story of patience, of transformation, of things quietly returning to where they came from so that life may begin again. When you hold a mushroom in your hand, you are not holding the whole organism, only the brief, delicate fruit of a vast hidden being. Beneath the ground lies a web called mycelium, a fine, silken network that threads through the soil like veins of thought. It is from this network that the mushroom rises, just as a flower blooms from its stem. But while flowers bloom to spread pollen through the air, mushrooms bloom to scatter spores, millions of them, drifting invisibly to start life anew elsewhere. The mushroom is the voice of the underground, the fleeting whisper of something much larger, older, and wiser than we usually imagine. If you could shrink down and walk through the soil, you would find a city beneath your feet, a shimmering web connecting tree roots to each other, carrying messages, trading nutrients, balancing the forest's needs. This is the quiet intelligence of fungi, a cooperation older than language, older than humanity, older than the first forests that ever grew. They do not move, but they reach. They do not speak, but they communicate. And through that stillness, they shape the living world. Tonight, we'll listen to their story, the science, the mystery, and the subtle beauty of these humble organisms that turn death into life. We'll explore where they came from, how they live, and what they might still be teaching us. But before we begin our journey into the fungal kingdom, if you're already feeling a little more relaxed or quietly curious, I invite you to subscribe to The Gentle Scientist. It helps others find their way here too. And if you'd like, leave a comment below telling me what time it is where you are, what part of the world you're listening from, or just how you're feeling now. I love reading your messages. It's always heartwarming to know that we're all here together, scattered across time zones, connected by curiosity and the shared quiet before sleep. So now, let your breathing slow. Let your body sink a little deeper into wherever you're resting. Feel the weight of the day begin to lift, just slightly. Tonight, you don't need to do anything at all except listen and drift. Let's begin. Imagine a dark, damp woodland after rain. The air is cool and still. Leaves lie in a soft, rustling blanket over the soil. At first, everything seems quiet and unchanged. And then, almost imperceptibly, the forest floor pushes back. A pale button of flesh appears between the leaves, then swells, stretches, and opens like a tiny umbrella. You might glance at it and think, there's a mushroom. But what you're really seeing is a message, a signal, and just one brief moment in a much longer life. When we talk about mushrooms in everyday language, we're usually talking about the visible, fleshy, spore-bearing fruiting body of a fungus, the part that emerges above ground on soil, wood, or other decaying matter. Scientists often describe a mushroom as a kind of fruit of the fungal organism, the way an apple is the fruit of an apple tree. It is the structure designed to make and release spores, the tiny reproductive units that drift on the wind or hitch a ride on passing animals. So when a mushroom appears on a log or in your lawn, what you're seeing is the reproductive phase of a much larger being. For most of its life, the fungus lives quietly, hidden from view in a web of fine threads. When conditions are just right, temperature, moisture, food, the hidden body decides it is time to reach into the air and let its spores go. The mushroom is that reaching, that brief, delicate structure devoted to reproduction, called a fruiting body or sporocarp in biology. Mushrooms are not plants and not animals. They belong to their own kingdom, the fungi, an entire branch of life that hums along in the background of ecosystems all over the world. Fungi are not green. They lack chlorophyll, the pigment plants use to capture sunlight. They cannot make their own food by photosynthesis. Instead, they live more like quiet digesters, secreting enzymes into their surroundings to break down complex materials and then absorbing the dissolved nutrients back into their cells. Because of this lifestyle, fungi often take on roles that plants cannot. Some live as decomposers, gently returning dead leaves, fallen trees, and other organic matter to the soil. Others form symbiotic partnerships, wrapping themselves around plant roots and trading nutrients, minerals and water from the fungus in exchange for sugars from the plant. This is one of the reasons fungi had to be recognized as something fundamentally different from plants. Their structure, their cell walls, their way of eating and living all told a different story. If you picture a classic mushroom in your mind, you can probably see the basic structure: a cap, a stalk, and something underneath. The cap, or pileus, is the umbrella-like top. It protects the delicate spore-producing surface below. The stalk, or stipe, lifts the cap up into the air, away from the damp ground, so that air currents can carry spores farther. Underneath the cap is where things become intricate. Many mushrooms have thin, radiating plates called gills, or sometimes a surface dotted with tiny pores or covered in little teeth or spines. This underside region, the spore-bearing layer, is the true working surface of the mushroom's reproductive machinery. You can imagine those gills or pores as a forest of microscopic factories. Along them sit specialized cells that actually make the spores. These cells are miniature containers for genetic information, packaging the fungus's instructions for life into dust-fine spores that can be carried away by the slightest breeze. If you've ever tapped a mature puffball and watched it release a cloud, or seen a mushroom drop a fine powder onto a surface overnight, you've witnessed this spore world in action. Each spore, if it lands somewhere moist and suitable, can germinate, It swells, wakes up, and begins to grow a tiny, thread-like filament called a hypha. One filament becomes many, branching and weaving through the soil or wood. Over time, they knit together into a dense, interlaced network called mycelium, the true body of the fungus. The mushroom itself begins as a tiny knot within that mycelial web, a little aggregation of hyphae called a primordium or pinhead. At first, it's smaller than a grain of rice, hidden within the substrate or just beneath the soil surface. As conditions stay favorable, this pinhead swells into a button, a round, compact structure that already contains, in miniature, the future cap and stalk. Then, in what feels like an overnight transformation, it rapidly expands. Instead of producing lots of new cells, it mostly inflates the ones it already has by filling them with water. This is why mushrooms can appear so suddenly after rain. The mycelium has already built the blueprint. It just has to pump the structure full of water to lift it into view. If you could peel back the forest floor like a carpet, you would find that the real drama is taking place below. Mycelium spreads out as a vast, branching network of hyphae, each filament just a few micrometers wide. Together, these threads can form patterns that extend meters, even kilometers, through soil and decaying wood, sometimes making up one of the largest and longest-lived organisms on Earth. Through this network, nutrients are moved from where they are found to where they are needed, and growth can be directed in response to food, moisture, or damage. In this light, a mushroom is a bit like a brief appearance on stage. The mycelium is the actor's entire life. All the rehearsals, the quiet days at home, the trips and the meals and the long thoughts. The fruiting body is just the moment the curtain opens. It rises, performs its role, releasing spores, and then withers away. The mycelium remains, invisible and persistent, ready to try again. The mushroom you see today is only one verse in a much longer song. Across the world, the verses of that song come in astonishing variety. There are mushrooms with broad, flat caps, ones with tall, pointed spires, delicate parasols as thin as tissue, and sturdy brackets stacked on tree trunks like shelves. Colors range from plain browns and whites to bright oranges, reds, and luminous yellows. Some even glow faintly in the dark. Scientists have described thousands of mushroom species, with more still being discovered, and they span every niche, from species that are safely edible and even delicious, to others that are dangerously poisonous, from large and fleshy to tiny and brittle. Despite their differences in shape, color, and size, these fungi share a set of underlying roles that keep ecosystems functioning. Many mushrooms are the outward sign of fungi doing the slow work of decomposition, breaking down tough materials like wood, leaves, and other plant litter. Using their enzymes, they unlock nutrients held in complex molecules, turning dead matter back into forms that plants and microbes can use. Without fungi, forests would choke under their own fallen branches, and soils would gradually lose their fertility. Other mushrooms signal partnerships rather than decay. In mycorrhizal relationships, fungal mycelia wrap around plant roots and extend far into the soil, greatly increasing the root system's reach. The fungus gathers water and minerals and shares them with the plant. The plant, in turn, offers sugars produced by photosynthesis. From above, you might see only the trees and a scattering of mushrooms, but below, a delicate lattice is humming with exchanges, quietly supporting entire forests and grasslands. So, when you see a mushroom, whether it's a small brown cap in the lawn, a cluster of oyster-like fans on a trunk, or a familiar button mushroom on your dinner plate, it is just the visible punctuation at the end of a long, underground sentence written in threads of hyphae. Now, we're going to gently lower our gaze and let our imagination sink down through the leaf litter and into the cool, dark earth beneath those mushrooms, where the true body of the fungus quietly spreads and listens. Beneath every mushroom you see, there is something far larger, far older, and far more intricate: the mycelium. Mycelium is the vegetative body of the fungus, the part that lives day after day, season after season, in soil, wood, or other substrates. It is made of countless fine threads called hyphae, slender filaments that are usually only about 1 to 20 micrometers in diameter, narrower than a human hair by orders of magnitude. These hyphae grow mainly at their tips. You can imagine them as soft, patient fingertips, pressing forward into the spaces between soil particles, exploring, sensing, and branching. With each tiny push at the growing tip, the fungus extends itself into new territory, weaving an intricately branched network that can spread out over large areas underground. If you could shrink down to the microscopic world and look closely at one of these hyphae, you would see that it isn't just a simple thread. Its wall is a layered structure, built primarily from chitin, the same tough material found in insect shells, combined with glucans and proteins. These chitin microfibrils are cross-linked with other molecules to create a wall that is both strong and slightly elastic, resilient enough to push between soil grains and around tiny obstacles without bursting. Now, imagine not just one hypha, but thousands, even millions, all extending and branching, looping and crossing, forming what scientists describe as a fractal-like fiber network. Statistically, the arrangement is almost random. Filaments run in many directions, interconnecting and interweaving so that no single hypha carries the entire burden. In mechanical terms, this web can behave a bit like an open cell foam, a light, porous structure that distributes force and adapts when compressed, bent, or disturbed. Deep in the dark, this foam-like network gives the fungus a combination of flexibility and strength. It lets the mycelium infiltrate complex, irregular environments: crumbly soil, rotting wood, tangled roots, without breaking. Even when parts of the network are damaged by animals, drought, or shifting soil, the rest can often take over, bridging gaps and rerouting flows. You could think of the mycelium as a quiet underground transportation and communication system. Through its hyphae, the fungus can take up water, nutrients like nitrogen and phosphorus, and a wide variety of organic compounds, then move them to where they're needed most. In soil, resources are often patchy, rich in one place, poor in another. The mycelial network helps the fungus bridge those patches, shifting its internal flows over centimeters, sometimes meters, balancing growth in different regions like water, finding many paths downhill. If one part of the network taps into a nutrient-rich pocket, that region can supply distant parts that are searching in leaner soil. If another part is damaged, crushed, dried, or grazed upon, the network can reroute around the injury. Nutrients, water, and even stress signals can move along these threads, allowing the fungus to respond to the world not as isolated fragments, but as a coordinated whole. The story becomes even more intricate when the mycelium meets plant roots. Many fungi form mycorrhizal associations, intimate partnerships with plants in which fungal hyphae and roots intertwine. In these relationships, the hyphae attach to root surfaces and may push their way into the root's outer tissues. In some types of mycorrhiza, such as arbuscular mycorrhizae, the fungal hyphae penetrate the plant's cell wall, but do not pierce the cell membrane. Instead, they fold the membrane inward, creating a highly branched, tree-like structure within the cell space. This greatly increases the contact area between plant and fungus, like many tiny fingers interlocked. Across this interface, there is an exchange. The fungus delivers water and minerals, especially hard-to-get nutrients such as phosphorus and certain forms of nitrogen, from the soil to the plant. In return, the plant supplies the fungus with sugars and other carbon-rich compounds made by photosynthesis. The mycelium, with its delicate, far-reaching hyphae, can explore volumes of soil that roots alone could never reach. In doing so, it effectively extends the plant's root system, improving its ability to survive in challenging conditions. From another perspective, you might see these mycorrhizal networks as shared roads. Through them, the fungus and plant trade what each is best at. The plant gathers energy from sunlight. The fungus gathers nutrients from the dark. Their partnership shows how deeply life on Earth is built on cooperation and interdependence, even when it is quiet and invisible. The architecture of mycelium is highly plastic and adaptable. When conditions change, when nutrients appear or disappear, when parts of the network are damaged, when competition increases, the fungus can modify its branching patterns, thickness of hyphae, and density of connections. In rich zones, mycelium may form dense clusters. In poor zones, it may send out long, exploratory filaments. Over time, the network structure reflects its history, where it has found food, where it has been stressed, and where it has invested in stronger, more permanent roads. In this way, mycelium behaves a bit like a living map that redraws itself, highlighting pathways that work and fading those that do not. The fungus doesn't have a brain, but its network can still solve problems. How to reach food efficiently, how to bypass obstacles, how to recover from injury, using the simple rules encoded in growth, branching, and resource flow. In this way, mycelium behaves a bit like a living map that redraws itself, highlighting pathways that work and fading those that do not. The fungus doesn't have a brain, but its network can still solve problems. How to reach food efficiently, how to bypass obstacles, how to recover from injury, using the simple rules encoded in growth, branching, and resource flow. If you return once more to that quiet forest where we began, you can imagine the soil not as inert dust, but as a space filled with this hidden web. Between grains of sand and specks of clay, hyphae thread their way. Around roots, they coil and branch. Inside rotting logs, they lace the wood like white veins. Tiny, invisible currents of water, minerals, and signals move along them. The mushrooms above are just the occasional punctuation marks, the visible signs that this unseen system is mature enough and well-fed enough to send up a fruiting body and reach into the air. Mycelium, then, is not just part of the fungus. It is the foundation of its life. It is the body that feeds, the network that explores, the web that partners with plants. It enables fungi to decompose, to recycle, to share, and to adapt. And in doing so, it quietly shapes the ecosystems we see: forests, grasslands, gardens, even the soil beneath our cities. As we rest for a moment, with that hidden web of mycelium in mind, those pale threads slipping between stones and roots, it's easy to forget that the same quiet strategy of growth can lead to very different shapes above ground. Some fungi create organisms so large that they rival entire cities in area. Others produce fruiting bodies so small that you could pass them by, mistaking them for dust or a wrinkle in a leaf. All of them are expressions of that same basic plan, a network of hyphae gathering resources and deciding at just the right moment to reach into the air. In the forests of eastern Oregon, there is a fungus that people affectionately call the humongous fungus. It isn't a fantasy creature or a mythic beast. It's a very real individual of Armillaria ostoya, a honey fungus whose mycelial network quietly spreads beneath the soil of Malheur National Forest. When scientists traced its genetic fingerprints from tree to tree, they found something astonishing. The same individual fungus, the same genetic clone, extended over more than 3.4 square miles, about 8.8 square kilometers. That's roughly 2,385 acres of forest held invisibly by one organism. By comparing its growth rates with its current spread, researchers have estimated that this ancient being may be thousands of years old, possibly between 2,000 and 8,000 years. Imagine that. While human civilizations rose and fell, this fungus continued to extend itself a few feet per year under fallen needles and stones through cool, dark soil. It doesn't hurry, it simply persists. Yet if you walk through that forest, you wouldn't see a giant. You'd see trees, grasses, patches of moss. And here and there, in certain seasons, you might notice small clusters of honey-colored mushrooms at the bases of infected trees, caps only a few centimeters across on modest stalks. Those are the fruiting bodies of Armillaria ostoyi, the visible moments in the long, formless story of its underground life. The scale of this organism is revealed not by a single towering mushroom, but by its mycelium and rhizomorphs, long, root-like cords of fungal tissue that creep under bark and along roots. Scientists mapped its spread by following these hidden structures and by sampling bits of tissue from many trees, checking their DNA, and finding the same individual over and over again. The mushrooms themselves are episodic, appearing in autumn when conditions are right, then collapsing back into the soil. The true bulk of the organism never shows its face. It lives in the wood, it decays, and the roots it infiltrates. A reminder that size in fungi is measured not only by what we see, but by what lies beneath. At the opposite end of the spectrum, there are mushrooms so delicate that an entire cluster could fit comfortably on your fingernail. Many species produce fruiting bodies just a few millimeters tall, with caps only 1 to 4 millimeters in diameter, roughly the width of a pinhead to the thickness of a pencil line. These tiny mushrooms often grow on individual leaves, pine needles, or the finest twigs, especially in humid forests where a thin layer of decomposing plant matter covers the ground. You might see them as tiny white or orange parasols on a fallen leaf, or as a scattering of translucent bells emerging from moss on a log. Under a magnifying glass, or in the close eye of a camera lens, these little mushrooms reveal a surprising complexity. Perfect gills, tiny stalks, sometimes even a faint glow in the dark in certain bioluminescent mycena species. But to the casual passerby, they're easy to miss. They live fast, drying quickly when the air does, and often rehydrating and reviving when rain returns. And beyond even these small fruiting bodies lie fungi too tiny to resemble mushrooms at all to our eyes. Microscopic structures, tiny cup-like fruiting bodies on wood, and unicellular fungi like yeasts, which are close relatives of mushroom-forming species but live as single cells. For these organisms, being small isn't a limitation, it's a strategy. It allows them to exploit thin films of moisture, minute pockets of nutrients, and surfaces we'd never even notice. Size isn't the only way fungi show their adaptability. Many species thrive in extreme environments where life is pared down to what is strictly necessary. In deserts, fungi survive with minimal water, forming small, slow-growing structures that hug the shade of rocks or live hidden within them. In Arctic tundra, filamentous fungi endure freezing temperatures and short growing seasons, adopting slow growth rates that match the harsh climate. In deep underground caves, some fungi persist with little light and limited organic matter, quietly recycling whatever reaches them. Bat droppings, plant bits washed in by water, the remains of insects. In these harsh conditions, a sprawling, fast-growing mushroom wouldn't make sense. Instead, fungi often remain small, compact, and patient, investing in resilience rather than spectacle. In all these places, the fungal body adapts. Hyphae can become thicker or thinner, networks denser or more sparse. Fruiting bodies, when they appear at all, are tuned to the environment, small, tough, sometimes only emerging during the brief windows when moisture and temperature line up just right. The size of a mushroom is shaped by many quiet forces working together. One is nutrient availability. To build a large fruiting body, the mycelium has to gather enough resources, carbon, nitrogen, minerals, and funnel them into a single structure. In rich substrates like large logs or long-lived tree trunks, it can afford to invest in massive polypores or thick clusters that persist for years. In sparse leaf litter or a thin twig, the fungus may only have enough energy to raise a tiny umbrella of tissue, just sufficient to release spores and vanish. Another factor is the type of substrate. Wood-decaying fungi that inhabit large tree trunks often produce sturdy, shelf-like fruiting bodies, while litter decomposers on leaves or needles grow small and delicate. The architecture of the fruiting body reflects the architecture of the food source, wide and heavy for big, solid pieces of wood, light and minimal for thin, scattered debris. Climate plays its part as well. In consistently moist, mild environments, mushrooms can safely grow larger and live longer without drying out or freezing. In places with frequent droughts or sudden temperature swings, ephemeral, small fruiting bodies that appear quickly after rain and disappear just as fast may be favored. And woven through all of this is evolutionary history. Each species carries a blueprint shaped by millions of years of adaptation to a particular niche, competition, and partnership. When we think of large mushrooms, we might imagine a classic cap and stem mushroom scaled up. But some of the largest fungal fruiting bodies look quite different. Many are bracket fungi, also called polypores, that grow in shelves or thick crusts on the sides of trees. These structures can become enormous, layered over years as the fungus continues to feed on the heartwood within. One species, Fomidoporia ellipsoidea from southern China, has been recorded with a fruiting body over 10 meters long and weighing an estimated 400 to 500 kilograms. A mushroom the size of a small car, hugging the trunk of its host tree. These large bracket fungi are like architectural outcrops of the fungal body, thick, woody, and long-lived compared to the soft, short-lived caps of many gilled mushrooms. While a tiny mycena might appear overnight and be gone in a day or two, a giant polypore can persist through seasons, a steady, patient presence slowly turning wood back into soil. All along this spectrum, mushrooms and their kin are working, decomposing fallen wood, recycling nutrients, helping plants gather water and minerals, surviving in deserts and tundra and caves. Size, in the end, is just one of the ways they adapt to the spaces they inhabit and the roles they play. Leaving behind the giants beneath the forest and the tiny umbrellas on single leaves, we can pause and notice something else. Even when mushrooms are the same size, they can look utterly different. Some are flame red and glossy, others soft brown and velvety. Some stand on tall, slender stalks. Others spread like shelves along a tree. In the quiet light of the forest, these differences are not random decoration. They are the shapes and colors that fungi have slowly chosen over millions of years to survive, to reproduce, and to speak silently with the world around them. If you picture a classic mushroom, you probably see a smooth cap perched on a stalk, with thin gills underneath. But this storybook mushroom is only one option. By stretching, compressing, or rearranging those basic parts, fungi create a whole gallery of forms. Some caps widen and fuse into brackets that fan out from tree trunks. Others abandon caps altogether and rise as coral-like branches, or form enclosed balls, puffballs, that keep their spores hidden until the right moment. The hymenophore itself can be flat gills, tiny round pores, folded labyrinths, or tooth-like spines. All of this sculpting has a purpose: to protect delicate tissues, to hold spores in the best position, to interact with wind, rain, and animals in just the right way. Then there is the matter of color. Many mushrooms are dressed in quiet earth tones, creams, browns, soft grays. Others, though, seem to have dipped themselves in a painter's palette. Scarlet caps, lemon yellow brackets, inky blues and purples, even greens and almost black. These hues are built from pigments, chemical compounds that absorb and reflect particular wavelengths of light. Fungi can produce melanins, which tend to create dark, protective colors. Carotenoids, which give yellows, oranges, and reds. Sometimes flavonoid-like and other phenolic compounds that add subtler tints. For the fungus, these pigments are not just for show. Dark melanins, for example, help shield cells from damaging ultraviolet radiation and oxidative stress. They can also make cell walls tougher, helping resist attack by microbes or grazing invertebrates. Carotenoid and phenolic pigments may also have antioxidant and antimicrobial roles, helping the fungus cope with light, drought, or infection. In this way, a mushroom's color can be a kind of invisible armor, a quiet chemistry of protection woven into its skin. Some mushrooms take color further, using it as a visual signal. In nature, bright, contrasting colors often serve as warning signs. Think of the red and white Amanita species that look like storybook toadstools, or the greenish caps of the death cap, Amanita phalloides. These mushrooms contain powerful amatoxins, molecules that can shut down essential processes in animal cells, and in the case of the death cap, are responsible for the majority of serious mushroom poisonings worldwide. Their colors, shapes, and sometimes distinctive odors may help warn off experienced foragers, both human and animal, who have learned, through evolution or misadventure, to associate those appearances with danger. Not all mushrooms broadcast themselves so boldly. For many species, especially those that live for months or years, camouflage is a better strategy. Bracket fungi that cling to bark can be mottled brown and gray, mirroring the tree's surface. Ground-dwelling mushrooms may be spotted in leaf litter colors, tan, umber, olive, so that they almost disappear among fallen leaves and twigs. For these fungi, blending in may reduce the chance of being gnawed by mammals, kicked by passing feet, or disturbed before their spores are fully released. The same pigments that provide physical protection can also serve as a visual cloak, letting the mushroom sink quietly into its background. Shape 2 is a language. A simple umbrella-shaped cap with gills underneath is actually a highly efficient spore distribution device. The gills dramatically increase the surface area where spores are produced, packing millions upon millions of them into a relatively small space. The spacing and orientation of the gills help create a still layer of air beneath the cap, allowing spores to drop cleanly before drifting into moving air at the cap's edge, where tiny currents can carry them away. Textures add another layer of adaptation. Some mushrooms are tough and leathery, their tissues dense and fibrous. These forms often grow on wood and may persist for months or years, enduring cycles of wetting and drying, cold and heat, insect chewing and falling branches. Their resilience allows them to keep producing spores over long periods. Others are delicate and ephemeral, made of thin, watery flesh that tears easily. These mushrooms appear quickly in moist conditions. After rain, during humid evenings, release a burst of spores and then wilt or vanish as soon as the air dries. Neither strategy is better. Each is tuned to a different rhythm of the environment. As you lie back and let your eyes grow heavier, you might imagine a soft, twilight wood after rain. Mushrooms of many colors and shapes dot the ground and cling to trunks, some glowing faintly in hidden logs, some nearly invisible against the bark, some bright and obvious as lanterns. Each one is shaped by centuries of evolutionary trial and error, tuned to its own way of surviving, attracting, warning, hiding, enduring. We were looking closely at mushrooms in the daylight, their caps and stems, their colors and textures, the ways they bend and twist to catch wind, hide from teeth, or call to insects with bright warning reds and soft camouflage browns. But when the sun slips away and the forest goes dark, some mushrooms stop using color altogether. They start using light. If you could walk slowly through a warm, damp forest at night and let your eyes adjust, you might notice something at the edge of your vision. A faint green shimmer on a fallen log, a ring of tiny lanterns nestled in moss, or thin, glowing threads tracing their way through rotting wood. These are bioluminescent mushrooms, fungi that make their own light, turning the forest floor into a quiet, living constellation. Unlike the way a white mushroom looks bright in a beam of moonlight, this glow doesn't depend on any outside lamp. It comes from inside the fungus itself, from a tiny chemical conversation going on in the dark. Scientists call this phenomenon bioluminescence, and in mushrooms, it happens because of a special pair of molecules, a light-emitting compound called luciferin and an enzyme called luciferase. When the mushroom is ready to shine, another enzyme, the fungal luciferase, often called luce, steps in. It helps transfer energy to the luciferin using oxygen and the cell's own energy currency. In the process, the luciferin is oxidized, releasing a small packet of energy in the form of a photon, a single particle of light. Because the fungus is alive and its metabolism is constantly ticking, this reaction can happen continuously, so long as there is enough oxygen and fuel. To our eyes, the result is a steady, soft glow, usually in the green range. All known bioluminescent mushrooms belong to certain lineages within the Basidiomycete order Agaricales, and across these species, the emitted light is remarkably similar in color, a cool, ghostly green that tends to look brightest to human eyes on moonless nights. This is very different from phosphorescence or fluorescence, which you might know from glow-in-the-dark stickers or objects that only shine after being charged with light. Those materials absorb light energy and then release it slowly, like a sponge being squeezed out. Bioluminescent mushrooms don't need to be charged. They don't store light from the day. Instead, they make light as a byproduct of real-time chemistry. It is an active process, powered by their own metabolism. When their biochemistry stops, if the tissue dies or dries out, the light goes out too. Bioluminescent mushrooms are rare. Out of more than 100,000 described fungal species, only a little over 100 are currently known to glow. So, why would a fungus, living quietly on rotting wood, spend precious energy to glow in the dark? One leading idea is that it's a kind of signal, and that the intended audience is not us, but insects. For a fungus, spores are everything, the future scattered on the wind. Many mushrooms rely on air currents alone to disperse them. But in dense forests, where wind is muted near the ground, attracting small animals that can brush against gills or caps or carry spores on their bodies adds another pathway for spreading. A glow that stands out in the dark, precisely when nocturnal insects are active, may give a subtle but important advantage. Not all glowing fungi follow the same script, though. In some Mycena species, the entire tiny mushroom, cap, gills, and stalk may emit light, creating miniature lanterns scattered over the forest floor. In others, only specific parts glow: the gills, the stem base, or the threads of mycelium hidden in wood. Different patterns of glow may reflect different roles. A shining cap might entice insects to land directly on the spore-bearing surface, while a glowing log threaded with luminous mycelium might deter certain grazers, attract predators of insects that feed on the fungus, or simply be a side effect of active metabolism in tissues busy digesting wood. What is clear is that, wherever they occur, these fungi are part of the machinery of decomposition and nutrient cycling. They grow on fallen logs, leaf litter, and rotting stumps, carrying out the slow work of breaking down lignin and cellulose and returning their building blocks to the soil. Their light may gently outline the places where this invisible recycling is strongest, tracing the shapes of logs and roots that are turning back into forest floor. To the ecosystem, the glow is incidental. To us, it looks like magic. That quiet magic has captured scientists' attention in another way too, not just as a curiosity to explain, but as a tool to borrow. Once researchers identified the key pieces of the fungal bioluminescence pathway, they realized they could move those genes into other organisms. In 2018, a team showed that by inserting the fungal genes into plants, it was possible to create autonomously glowing plants, ones that don't need external chemicals, but produce their own light from their everyday metabolism. Since then, researchers have refined and optimized this pathway, boosting brightness and stability in different hosts, including plants and mammalian cells. Most recently, you might have seen stories about the firefly petunia, an ornamental plant engineered to carry genes from bioluminescent mushrooms, glowing softly green along its veins and flowers, its brightness following the rhythm of growth and metabolism. These plants are part artwork, part experiment, living demonstrations of fungal chemistry at work in leaves and petals instead of caps and gills. Outside the lab, bioluminescent mushrooms have also seeped into culture and imagination. Photographers hike into forests to capture the faint glow of Mycena chlorophyllus in Asia or ghost fungi in Australia. Artists and writers use glowing mushrooms as symbols of hidden life and wonder, small, steady lights in wild, forgotten places. The Swiss Federal Institute for Forest, Snow, and Landscape. Research has written about how these fungi intrigue both artists and scientists, bringing people into the woods at night to look more closely at decaying logs they might otherwise ignore. In stories and illustrations, bioluminescent mushrooms become waymarks in fantasy forests, places where travelers rest or meet strange companions. In reality, they are quieter than that. They do not pulse or flash like fireflies. They simply shine, slowly, constantly, like the dim light in a child's night lamp. Still, we're going to stay in that darkness, but bring in a different sense. We're going to talk about a fungus that communicates not with light or color or visible form, but with scent, so powerful and strange that it can pull bears to their feet, make elk dig until their noses bleed, and persuade humans to pay thousands of euros for a few small pieces shaved over dinner. We're talking about truffles. If you've ever caught the true smell of a fresh truffle, you know that it doesn't behave like an ordinary odor. It doesn't just sit politely in the background the way a mild mushroom or a loaf of bread might. It arrives with a kind of insistence, sharp, strong, and faintly luxurious, pushing its way up into your awareness. People reach for all sorts of metaphors to describe it: earthy, garlicky, musky, even a little wild. In fine restaurants, that smell is treated almost like a performance. A small, knobbly truffle appears at the table. The server holds a thin metal slicer. A few delicate shavings fall onto pasta, or eggs, or risotto. The truffle itself may look modest, almost like a small, wrinkled stone, but the price is anything but modest. Because they are rare and difficult to find, 2 kilograms of Piedmont white truffles can cost around 12,000 euros. People pay not for the way they look, but for that fleeting, unforgettable aroma. For most of the year, though, truffles are not on linen tablecloths. They are not even really truffles in the sense we think of them. Most of the time, they are simply mycelium in the soil. The dense, rounded truffle that humans eat is a fruiting body, a reproductive structure. It's the fungus's way of packaging spores. But unlike the mushrooms that lift their caps into the air, truffles develop entirely underground. They never break the surface, never fan out gills to the wind, and that creates a problem. To reproduce, fungi need their spores to go somewhere new, to be carried away from the parent body, so they can germinate in fresh territory. Many mushrooms solve this by releasing spores directly into the air. Gravity and breezes do the rest. Others hitch rides on insect bodies or raindrops. But if your fruiting body is buried in the soil, the air doesn't come to you so easily. The truffle spores are safe underground, but also stuck. So truffles have evolved a very different strategy. They don't push their bodies upward. They pull the world downward instead. They do this with smell. When the time is right, when the mycelium has formed a truffle that is mature and ready, these underground fruiting bodies begin to release a complex cocktail of volatile chemicals. This is the truffle aroma as we know it on the plate, but its original audience is not us. It is the noses of animals. For a truffle, scent is a kind of rope thrown up to the surface, a way of saying, "I'm here, dig me up." The fungus cannot walk or climb. It cannot lift itself into the wind, but it can reach up with molecules, letting them diffuse through the soil until they find a sensitive nose. That nose might belong to a wild boar, which snuffles and digs, ingesting truffle tissue and later spreading the spores elsewhere. It might belong to a fox, or a small mammal, or even to humans with trained dogs and pigs. The sooner a ripe truffle is found and unearthed, the sooner its spores can travel. The smell is an invitation, and also of a deadline, a signal that says, now is the time, come quickly. Over long stretches of evolutionary time, this scent has been shaped by the preferences of animals. It is no accident that it smells so compelling. In the forests where truffles grow, bears have been known to hurl aside fallen tree trunks, driven by the smell of ripe truffles below. Elk dig with such enthusiasm that their noses sometimes bleed. The truffle's fragrance reaches into mammalian brains and tugs, triggering curiosity, hunger, and sometimes something more. Humans, too, seem to sense something deeply animal in that aroma. We so often link truffles with desire. To us, the scent is luxurious. To the truffle, it is simply effective. It convinces large animals to go to the trouble of digging through soil, damaging their hooves or snouts, just to reach a buried lump of fungal tissue. But the truffle's powers of attraction don't begin at the surface. Long before a boar or a chef ever notices its presence, the fungus has already been practicing a more subtle kind of invitation. The story starts with hyphae, those narrow threads that fungi are made of. At first, truffle hyphae are small, separate strands in the soil, spreading and probing, much like other fungi. They reach out into the environment, and when compatible hyphae meet, there is an opportunity. Chemical signals, almost like pheromones, can draw them together, encouraging them to fuse and combine their genetic material. In this way, hyphae from different individuals can form a shared mycelial network. What begins as scattered threads gradually becomes a more organized web, a kind of fungal body made up of countless hyphal tips. The truffle that will eventually be unearthed is the final product of this gathering, a physical expression of mycelia that have successfully recognized each other, merged, and prepared to reproduce. You could think of this as the fungus's first invitation. Hyphae charming other hyphae, using microscopic cues to invite union and exchange. There is no courtship dance in the way animals do it, no song, no display of feathers, but there is a conversation in chemicals. Come closer, we are compatible. Let's build something together. Once a mycelial network has formed, another invitation must take place. Because the truffle fungus cannot produce all the carbon-rich compounds it needs by itself. Like many other fungi, it depends on a plant partner. Above the truffle, in the sun, live trees and shrubs. They capture light and turn it into sugar through photosynthesis. Below the truffle, in the dark, the fungus is very good at exploring soil, unlocking nutrients like nitrogen and phosphorus from complex organic matter. A partnership is possible, but it doesn't happen automatically. It begins with another chemical courtship. Plant roots release a variety of compounds into the soil, small molecules that leak or are actively exuded into the rhizosphere, the microscopic world around the root. Some of these compounds act as signals. When truffle spores in the soil detect the right kinds of root-derived chemicals, they are nudged into action. They germinate more readily. Their hyphae grow faster, branching more, extending farther. At the same time, the fungus sends out its own signals in return. Fungal compounds can cause plant roots to change their shape, to form more fine, hair-like branches. tiny root tips that explore more soil volume. This increased branching raises the odds that a root tip and a fungal hypha will encounter each other in just the right way. Roots and hyphae do not see each other, they do not hear each other, but they can feel each other chemically. The plant says in its own molecular language, "I'm here, I could be a partner." The fungus replies, "So am I, let's meet." Each side reshapes itself, guided by signals it cannot think about but can respond to. Eventually, a root tip and fungal hypha make contact. The fungus wraps around the outer layers of the root or slips between root cells, forming a mycorrhizal association. From that point on, sugars from the plant can flow into the fungus, and nutrients and water from the fungus can move into the plant. The truffle's mycelium now has the carbon it needs to grow, and the tree has a more efficient way to explore the soil. Only after this partnership has matured, after the underground network is well fed and stable, does the fungus invest the energy to form a truffle fruiting body. By the time scent enters the story, the truffle's life has already passed through many quiet stages of attraction and union, all of them invisible. When a truffle is young and still developing, it is relatively modest. Its aroma is faint, not yet ready to call the world to its side. Inside, spores are maturing. The tissues are thickening. The fungal cells are arranging themselves into the intricate chambers that will hold the next generation. Then, when conditions are right, season, moisture, temperature, the state of the mycelium and its partner roots, the truffle begins to craft its signature fragrance. This scent is not a single molecule, but a blend. Dozens, even hundreds of volatile compounds, each with its own note, mixed in specific proportions. The exact recipe depends on the species of truffle, the soil it grows in, the climate that shapes that soil, and the diverse microbial communities that share its underground home. Bacteria and other fungi living on and within the truffle can modify its chemistry, adding or changing odor molecules. The result is that each type of truffle, white, black, summer, winter, has a characteristic perfume, and even truffles of the same species from different regions can smell subtly different. This is why people speak of terroir for truffles, just as they do for wine, a reminder that the fragrance you experience is not just the work of the fungus, but the work of an entire community of earth, climate, and microbes. To animals, this fragrance carries information, not just food, but food here, now, ripe. To us, it is mysterious, hard to describe but hard to ignore. Most of the rituals of truffles are never noticed by us. We do not see hyphae recognizing each other. We do not see spores waking up in response to root exudates. We do not see roots changing shape under the influence of fungal compounds. We arrive late to the story. By the time a truffle's smell is strong enough for a human to notice, the invitation has already been written and sealed. We are not the main characters. We are just one more species with a nose, one more animal drawn into an ancient agreement between fungus and the world above it. A buried fungus has reached up and gently tugged at distant minds using nothing but molecules and patience. It's easy to think of truffles as luxury ingredients, little treasures shaved over expensive plates. But from the fungus's point of view, They are something else. Chemical letters addressed to the entire animal world, written in a language of scent, asking for one simple favor: find me, eat me, carry me onward. And when you next see or smell a truffle, even just in imagination, you can remember that behind that moment is a hidden drama of attraction and partnership, all of it happening slowly, quietly, under the surface. Yet these mushrooms are only the visible signs of something much larger, a deeper, older conversation, one that has been going on for hundreds of millions of years. It's a conversation between fungi and plants, carried not by sound, but by sugars and minerals, by genes and chemicals, flowing through roots and mycelium in the dark soil. This kind of partnership is called symbiosis, a close, long-term relationship between different organisms. In the case of fungi and plants, that relationship is often mutualistic. Both partners benefit. For most of the plants you see on land, from forest trees to meadow flowers, this partnership isn't a luxury. It's a foundation. Scientists estimate that around 80,000 to 90% of land plant species form symbiotic associations with fungi known as mycorrhizae, helping them obtain nutrients and water, and shaping entire ecosystems in the process. The word mycorrhiza simply means fungus root. Under the microscope, it describes an intimate intertwining of plant roots and fungal hyphae. You can imagine these two styles as different ways of holding hands. One grips the root from the outside, weaving between its cells. The other reaches into the root cells, but very carefully, stopping at the membrane and creating an intricate handshake surface inside. Both arrangements create a gateway through which nutrients and water can flow between partners. Through these gateways, a quiet trade takes place. Fungal hyphae extend far beyond the plant's own roots, exploring volumes of soil that root hairs alone cannot reach. They are especially good at capturing phosphorus, a key nutrient that often binds tightly to soil particles, as well as forms of nitrogen, potassium, and trace minerals. They also help plants tap into thin films of water held between soil grains, making moisture more available during dry periods. In return, the plant sends the fungus something the fungus cannot make for itself: carbon-rich sugars produced by photosynthesis in leaves. These sugars travel down through the plant's vascular system and are released to the fungus at the root interface. In many cases, plants may allocate 10 to 30% or more of their photosynthetically fixed carbon to their fungal partners. A substantial investment, but one that pays off in improved nutrient and water supply. This means that when sunlight touches a leaf high in the canopy, some of that light's energy eventually ends up at the very tips of fungal hyphae deep in the soil. In the other direction, phosphorus released from mineral particles or organic matter by fungal enzymes can end up powering reactions in the plant's chloroplasts. The fungus and the plant are, in a sense, sharing one metabolism stretched out across two bodies. From the plant's perspective, the fungus effectively extends its root system. Instead of roots alone, the plant now has roots plus a web of fine fungal threads reaching into tiny spaces and distant patches of soil. This is particularly important in nutrient-poor or dry environments, where every extra millimeter of reach can matter. Trees in boreal forests, grasses in poor soils, shrubs on dry hillsides. Many of them owe their health, and sometimes their survival, to fungal partners that help them drink and feed more efficiently than they could alone. But such intimacy is risky. From the plant's point of view, letting a fungus grow into or around its roots is a delicate decision. The fungus must offer enough benefit to justify lowering its defenses. To manage this, plants and fungi engage in biochemical and genetic conversations before and during colonization. When fungal spores germinate near roots, they release signaling molecules into the soil. The plant, in turn, exudes its own set of compounds, often including special lipids and small organic molecules that the fungus can detect. If the plant recognizes the fungus as a potential partner rather than a pathogen, it activates a shared internal pathway. This pathway coordinates changes in gene expression that open the door to colonization. The plant adjusts its immune system, restructures its root cells, and prepares to accommodate hyphae. The fungus shifts its growth to form specialized structures instead of simply invading and destroying tissue. This careful regulation ensures that the relationship stays in balance, mutually beneficial rather than parasitic. Plants can also scale back or terminate symbiosis if conditions change, if soils become richer, or if the fungus takes too much and returns too little. The partnership is always being renegotiated, molecule by molecule. The story of this partnership stretches far back into deep time. Fossil evidence and genetic analyses suggest that mycorrhizal symbioses arose very early in the history of land plants, on the order of 450s, 500 million years ago, around the same time that plants were first colonizing land. In ancient fossil deposits like the Rhynie Chert of Scotland, scientists have found preserved symbiotic structures inside the tissues of early plants, clear signs that fungi and plants were already cooperating to share nutrients. Many researchers now think that fungi were crucial in making the move onto land possible. Before plants evolved complex root systems, the earliest land plants would have struggled to extract nutrients such as phosphate from bare mineral surfaces. Fungal partners, able to dissolve minerals and reach into tiny cracks, may have acted as primitive root systems, helping those first plants survive and spread. Some of the most delicate and specialized examples of this dependence can be seen in orchids. Orchid seeds are astonishingly small and almost dust-like, containing little or no nutrient reserve. On their own, they cannot germinate and grow into seedlings. Instead, they rely on mycorrhizal fungi to invade the seed and supply carbon, water, and minerals during early development. Only with a fungal partner feeding it from the inside can an orchid seed awaken and become a tiny plant. This reliance on fungi can continue in varying degrees throughout the orchid's life. Other plants benefit in different ways. Some mycorrhizal fungi appear to protect their hosts from pathogens, either by directly competing with harmful microbes around the roots or by priming the plant's immune system so it responds more effectively. Still others help plants cope with environmental stresses like drought or heavy metals, moderating the flow of water and adjusting the chemistry of the soil immediately around the roots. When you zoom out from a single plant, the picture becomes even more intricate. Mycelial threads do not stop at one root system. They often connect multiple plants together, forming what researchers call common mycorrhizal networks, sometimes poetically nicknamed the Wood Wide Web. Through these shared fungal networks, resources like carbon, nitrogen, and phosphorus can move between different plants, sometimes from older, well-established trees to younger seedlings growing in their shade. If you imagine a forest now, you might see it differently. Not just trunks and leaves and individual root systems, but a shared underground web. Fungal threads touching root tips of many species at once, forming a living mesh. Sugars flow down from sunlit crowns into this mesh. Minerals and water flow up from the soil. Some plants give more than they receive at certain times. Others, like shaded seedlings, may receive more than they can yet pay back. Over years and decades, this give and take helps shape which trees thrive, which saplings survive, and how resilient the whole community is to storms, droughts, or disease. All of this is happening quietly all the time, whether anyone is there to notice or not. Mushrooms show us where these relationships are flourishing. They are small reminders that no tree stands alone. No plant is truly isolated, and that even in the stillness of the night, the living world is held together by countless unseen partnerships, working together in the dark while you rest and drift toward sleep. But plants are not the only ones listening to fungi. All through the living world, animals are paying attention too. In the hush of the forest, mushrooms are not just recyclers and partners. They are also food, soft, fragrant, nutrient-rich food for a surprising number of hungry mouths. If you picture a mushroom in the wild, it might be easy to imagine it standing alone, untouched. In reality, many fruiting bodies are bustling feeding stations. From tiny insects to squirrels, deer, and even bears, animals weave mushrooms into their diets. And in doing so, they become part of the fungus's life story as well. If we shrink our point of view down to the forest floor, to the level of moss and leaf litter, the first mushroom eaters we meet are often invertebrates. A fresh mushroom cap can be alive with minute visitors, flies, fungus gnats, beetles, mites, springtails, nematodes, and more. Many of these animals feed not only on the flesh of the mushroom, but also on its spores and mycelium, tunneling through the soft tissues, scraping gills, or grazing the underside of the cap. Some flies lay their eggs directly on or inside the fruiting body. When the eggs hatch, the larvae begin to burrow and feed, turning the mushroom into a moist, sheltered nursery. Beetles and their larvae do the same, carving intricate galleries through the flesh. Springtails, tiny, agile creatures that live in the damp spaces between soil particles, may nibble on spores and surface tissues. To us, the mushroom looks like a single object. To these small creatures, it is a habitat, food source, and meeting place all at once. As they come and go, these invertebrates do more than just feed. Spores cling to their legs, their bodies, sometimes pass through their guts, and emerge still viable. Researchers have found that animals can disperse living fungal spores after eating fungi, carrying them from one patch of forest to another. For some species, such as truffle-like fungi that fruit underground, this animal-mediated dispersal is not just helpful, it is essential. Over time, some insects have gone beyond opportunistic snacking and become specialists. Certain beetles, flies, and even ants rely heavily or exclusively on fungi as their main food, and in doing so, they've had to solve a tricky chemical problem. Many mushrooms defend themselves with toxins. Studies on mushroom-feeding fruit flies, for example, show that some lineages have evolved tolerance to cyclopeptide toxins found in deadly species like Amanita phalloides. These insects carry genetic and biochemical tools that let them survive doses that would be lethal to other animals. An evolutionary adjustment to a fungal menu. This is a quiet arms race, played out in molecules rather than claws and teeth. Fungi evolved defensive compounds to discourage being eaten. Insects evolved detoxification enzymes, altered receptors, or storage mechanisms that neutralize or compartmentalize those same compounds. The result is a delicate balance. The fungus is not entirely safe, but the specialist insect gains a rich, relatively competition-free food source. If we pull back to a larger scale, we begin to see small mammals joining this fungal feast. Rodents, such as squirrels, chipmunks, voles, and mice, are well-known mycophages. In some forests, analyses of stomach contents and feeding behavior show that fungi, especially underground truffle-like forms, can make up a major portion of their diet at certain times of year. For these animals, mushrooms and truffles offer carbohydrates, proteins, and micronutrients, sometimes even serving as an important fallback food when seeds or fruits are scarce. Many larger animals also include mushrooms in their diet when they come across them. Deer, for example, will browse on fleshy fungi in woodlands, adding them to their menu alongside leaves and twigs. Bears, especially in forests rich with fruiting fungi, have been observed eating mushrooms as a seasonal food, gaining energy and nutrients from the dense flesh. Some primates, including certain monkeys, are known to consume fungi as part of a varied, opportunistic diet. For these larger animals, mushrooms are often one food among many, but they can become particularly valuable during specific seasons, such as late summer or autumn in temperate forests, when fungal fruiting peaks and other resources may be declining. Whenever an animal eats a mushroom and moves on, it carries the fungus's story with it. Spores that survive the journey through the digestive tract emerge in feces, often deposited far from the original fruiting site and packaged in a little pellet of fertilizer. For underground fungi that cannot shoot their spores into the air, this is the main way they travel. Even for above-ground mushrooms, mycophagy by animals can supplement wind dispersal, scattering spores into new habitats and helping maintain genetic diversity across landscapes. In this sense, mycophagous animals and fungi are often engaged in a kind of mutualism. The animals gain food, and the fungi gain transportation. Not every bite is helpful. Some fruiting bodies are shredded before spores mature, and some spores are destroyed in digestion. But across whole ecosystems and long spans of time, animal movement contributes significantly to fungal spread and persistence. These feeding relationships ripple outward into broader ecological patterns. When small mammals rely on fungi as a major food source, their populations can rise and fall with fungal fruiting cycles. Those mammals, in turn, are prey for predators like owls, foxes, and mustelids. So a good mushroom year doesn't just feed the squirrels, it may indirectly support higher survival for the predators that hunt them. By influencing animal nutrition and behavior, fungi become quiet architects of population dynamics and food web structure. In some forests, the abundance of certain mycorrhizal fungi, the populations of small mycophagous mammals, and the health of trees are all intertwined in feedback loops. Fungi feed mammals, mammals disperse fungi, fungi nourish trees, and trees provide habitat for both fungi and animals. Of course, not all fungi are eager to be eaten. Many produce potent toxins or psychoactive compounds, which likely act, at least in part, as defense mechanisms against herbivory. Deadly peptide toxins, such as those in some Amanita species, make a single bite a serious risk for vertebrates. Other compounds, like the psychedelic alkaloids in psilocybe mushrooms, may alter the behavior of insects and other small animals in ways that discourage repeated feeding. Here again, evolution has staged its slow arms race. Fungi invent new biochemical tricks. Animals evolve ways to avoid, tolerate, or even exploit them. Some insects specialize on toxic mushrooms, turning a dangerous resource into a private buffet. Others learn, through instinct or experience, to avoid certain colors, scents, or tastes that signal trouble. Now we're going into the human story, into kitchens and temples and old stories told by firelight, and see how mushrooms have quietly followed us through history. Because for us, fungi have never been just background biology. They have been food, medicine, omens, and symbols. They sit in the center of fairy rings in children's books, glow in paintings and tapestries, and simmer in soups that smell like home. They are woven into myths about gods and spirits, and into modern festivals and pilgrimages. If you trace human culture gently with your fingertip, you find little mushroom-shaped imprints everywhere. Long before modern supermarkets, people were already bending down to pick mushrooms from the forest floor. In ancient Rome, cooks and writers recorded their fascination with wild fungi. The Roman cookbook, attributed to Apicius, compiled around the 1st century CE, includes recipes using mushrooms as delicacies, ingredients that signaled refinement, luxury, and the pleasures of the table. Later, historical work suggests that species like oysters, morels, and other prized fungi found their way into imperial kitchens, served to guests as a sign of status and connection to nature's subtler flavors. Across the Mediterranean, mushrooms flicker in and out of the record, sometimes exalted, sometimes feared. In some periods, they were regarded as food of the gods. In others, they were treated with suspicion, their mysterious powers of life and death too close for comfort. In East Asia, courtly cuisines elevated certain fungi, such as matsutake and shiitake, into seasonal treasures reserved for nobility or special occasions, a tradition that still echoes in the high prices and rituals surrounding them. Humans didn't just eat mushrooms, we painted with them. Historical and popular accounts describe how Roman artists experimented with pigments and dyes derived from fungi, and some modern writers point to wall paintings in places like Herculaneum, buried by the eruption of Vesuvius in 79 CE, as among the earliest sites where mushroom-based dyes and paints may have been used. Whether or not every example holds up under modern chemical analysis, the idea itself of humans grinding fungi into color, spreading them in thin layers over plaster, captures the same quiet fascination we see today in mushroom dyers who coax golds, reds, and browns from chanterelles and bracket fungi. Over time, mushrooms slipped from the kitchen and workshop into the realm of the sacred. In Mesoamerica, long before European contact, several cultures, including the Aztec and Maya, used psychoactive mushrooms in rituals and healing practices. In these ceremonies, led by skilled healers or shamans, the mushrooms were not a casual pastime. They were a structured, spiritual technology, a way to enter a space where messages from gods, ancestors, or the spirit world might be heard. On the African continent, ethnomycological studies and oral traditions hint at rich histories of mushroom use, edible, medicinal, and visionary, among various peoples. But colonization, cultural disruption, and the passing of elders have left many of these practices only partially recorded. Modern researchers working with communities in Africa and elsewhere are now racing gently against time to document traditional knowledge of fungi, which species are eaten, which are avoided, which appear in stories or rituals, and how people understand their roles in the local cosmos. Wherever mushrooms and humans meet, stories seem to sprout. In European folklore, rings of mushrooms in a meadow became known as fairy rings, places where it was said that fairies danced at night, leaving behind a circle no human should step into lightly. Entering such a ring might mean being lured into the fairy realm or cursed with misfortune. In other tales, it promised good luck or secret knowledge. Red-capped toadstools with white spots, Amanita muscaria, appear under the feet of gnomes and elves in illustrations, decorating children's books and postcards, blending real chemistry with imagined magic. Today, mushrooms continue to glow at the edges of modern culture. They appear in fantasy novels, video games, and films as familiar shorthand for enchanted forests and whimsical realms. They inspire architects and designers who build mushroom houses or use fungal forms as motifs. They color fabrics and yarns in natural dye studios. Online, artists share paintings and prints of fairy rings, ink caps, and coral fungi. Foragers post baskets of chanterelles and porcini as if displaying treasure. Articles, podcasts, and exhibitions talk about mushroom culture as its own phenomenon, a blend of gastronomy, environmentalism, spirituality, and art. Alongside this aesthetic fascination, mushroom tourism has emerged. Travelers journey to forests in Japan, hoping to taste wild matsutake, or to Eastern Europe to walk ancient mushroom trails. Others seek out workshops with local experts, learning how to gather safely and respectfully. In some regions, long-standing mushroom festivals celebrate the autumn harvest, featuring cooking competitions, identification walks, and storytelling sessions that pass on folk knowledge to a new generation. Through all these threads runs a simple fact: mushrooms are, at the same time, ordinary and extraordinary. In one moment, they are tonight's dinner, sliced into a pan with garlic and oil, part of daily sustenance. In the next, they are holy objects placed on an altar, or tiny painted toadstools in the corner of a medieval manuscript, or cautionary figures in a tale warning children not to stray too far into the woods. The same kingdom of organisms feeds us, heals us, inspires us, and sometimes frightens us, depending on the story we choose to tell. As your eyes grow heavier, You might imagine a single mushroom in a clearing at dusk. To a squirrel, it is food. To a beetle, it is shelter. To a mycologist, it is a data point. To a child, it might be a fairy stool or a tiny house. To an elder in a mountain village, it might be a medicine taught by their grandparents. To a pilgrim in a distant land, it might be a key to the sacred. Now, just for a little while, let's let that ground fall away. Imagine the sky above you darkening, stars brightening, and a small vessel drifting silently through the night. Inside, a handful of humans are trying to carry a living world with them. And somewhere in that tiny, floating ecosystem, there may be mushrooms. It sounds like science fiction, but scientists are already exploring how fungi might travel with us beyond Earth. In laboratories, on the International Space Station, and in design studios, researchers are asking a quiet, astonishing question. Could fungi help us survive and build in places where there is no soil, no forests, and only thin air, or none at all? Part of the reason anyone dares to ask is that fungi have already proven themselves to be remarkably tough. Some species, especially those with dark melanin pigments, can survive intense radiation, deep cold, scarcity of nutrients, and cycles of drying and re-wetting that would kill many other organisms. After the Chernobyl nuclear accident, scientists discovered black melanized fungi colonizing the damaged reactor walls, apparently not just enduring radiation, but responding to it with enhanced growth. Fungal spores, protected by sturdy cell walls and pigments, can remain viable after exposure to harsh conditions. And fungal colonies have even been found on the surfaces and equipment of space stations, adapting their morphology to microgravity and the confined artificial environment. To engineers and life support designers, this resilience looks like an invitation. A long-duration space mission, or a future base on the Moon or Mars, must function as a closed ecological system, where air, water, and nutrients are recycled as completely as possible. Every bit of waste, inedible plant stems, packaging, worn textiles, even certain plastics, must be handled carefully. On Earth, fungi excel at this kind of work. Their mycelium infiltrates wood, leaves, and other organic matter, releasing enzymes that chop complex molecules into simpler ones that can be reused by plants and microbes. In space, that same ability could help close the loop. Projects like Melissa, Microecological Life Support System Alternative, explore microbe-based ecosystems that recycle gases, liquids, and solids to support human crews. While many focus on bacteria and algae, fungi fit naturally into this picture as decomposers, turning cellulose-rich waste, inedible crop residues, and perhaps even some synthetic materials into simpler compounds, while producing useful biomass in the process. Envision a lunar or Martian habitat where bags of straw, plant trimmings, or worn cloth are colonized by mycelium, gradually transformed into both edible mushrooms and nutrient-rich material that can re-enter the system. That leads to another role: food. Mushrooms are compact, high-yield crops. They don't need sunlight in the way green plants do. They require controlled temperature, humidity, oxygen, carbon dioxide, and suitable substrates. They're rich in protein, B vitamins, and minerals. Some species can also supply vitamin D when exposed to ultraviolet light, something astronauts normally receive as supplements. Recently, experiments like mission mushroom have begun testing whether oyster mushrooms can fruit in microgravity. In early 2025, an Australian-led team sent kits containing mycelium and substrate aboard a SpaceX mission, aiming to see if mushrooms could complete their life cycle while orbiting Earth. Other studies are exploring mushroom cultivation on the International Space Station, sometimes using simulated waste streams like inedible plant biomass, textiles, and packaging as substrates. The vision is simple and elegant. Turn waste into food. In a closed spacecraft, anything that can be safely eaten is precious. If mycelium can grow on materials that would otherwise be discarded and produce nutritious mushrooms in a small volume, every gram of payload becomes more valuable. Oyster mushrooms, in particular, grow quickly and tolerate a variety of substrates, making them strong candidates for future space gardens. Fungi also offer biotechnological tools beyond food. Certain species can degrade complex pollutants and toxins, a property used on Earth in bioremediation to help clean up oil, pesticides, and industrial chemicals. In a spacecraft or off-world habitat, fungi might be used to help break down persistent organic wastes or to stabilize and detoxify materials before they are recycled. Some researchers even imagine engineered fungal strains designed to tackle specific chemical challenges in life support systems, quietly working in bioreactors while humans sleep. Then there is the idea that seems to come straight from a dream: building houses from fungi. NASA researchers and their partners are working on myco-architecture projects that explore using mycelium-based composites as construction materials for lunar or Martian habitats. Mycelium can be grown into molded shapes, panels, bricks, shells, by allowing it to colonize a matrix of agricultural waste, sawdust, or other fibers. Once it fills the mold and binds the material together, it can be killed or inactivated to halt growth, leaving behind a lightweight, surprisingly strong, and insulating composite. Tests on Earth show that well-designed mycelium materials can have compression strengths comparable to some types of lumber, flexural strengths rivaling reinforced concrete in certain directions, and useful fire resistance and insulation properties, all while being biodegradable and produced with low-energy inputs. NASA's concept imagines astronauts arriving on another world with lightweight shells or inflatable frames that serve as outer scaffolds. Inside those shells are dormant fungal spores and a dry substrate. Add water, control conditions, and the mycelium begins to grow, reinforcing the structure from within. Once the material reaches the desired density and strength, growth is stopped, and the habitat becomes a stable, solid shelter. In theory, such habitats could even be partially self-repairing. If microcracks or damage occurred, carefully managed mycelium might be encouraged to grow into the gaps. Fungi may also offer help with one of the most serious invisible threats in deep space: radiation. Those melanized fungi from Chernobyl, sometimes called radiotrophic fungi, appear able to use ionizing radiation as an energy source, or at least to tolerate it unusually well, thanks to the special electronic properties of melanin. Experiments have tested how well mats of such fungi can attenuate radiation, and some analyses suggest that fungal biomass rich in melanin could contribute to lightweight radiation shielding, either as a coating on walls or as layers within structural materials. In a future habitat, one could imagine walls composed of several layers, perhaps an external regolith shell, an inner mycelium-based composite, and within it, zones enriched with melanized fungal tissue, helping to soak up part of the cosmic radiation that would otherwise pass through metal or plastic. Such biological shields wouldn't replace other forms of protection, but they might reduce the mass of heavy shielding materials that must be launched from Earth. To move beyond imagination, of course, we need data. That is why different space agencies and research groups have started sending fungi, not just spores, but live cultures and mycelium, into orbit. Some experiments have focused on how microgravity and radiation affect fungal growth, morphology, and gene expression, while others look specifically at their performance in tasks like waste conversion or nutrient recycling. In 2024, for example, a Swinburne University-led experiment sent species like lion's mane, turkey tail, and cordyceps to the International Space Station to study how they adapt and what compounds they produce in microgravity. In parallel, concepts like Melissa continue to refine how to integrate fungi and other microbes into bioregenerative life support systems, where every breath of carbon dioxide, every drop of wastewater, and every scrap of organic material is part of a carefully balanced cycle. But the path is not simple. Growing fungi in space raises challenges that must be addressed with patience and precision. Microgravity affects how fluids behave, which in turn influences how mycelium gets oxygen and moisture. Waste substrates may behave differently when they are not settling under their own weight. Fungal spores are incredibly persistent. In a sealed craft, uncontrolled growth or contamination in the wrong place could create hygiene or material problems. Engineers must design systems that can contain and guide growth. Sealed bioreactors, well-characterized strains, and robust monitoring to ensure that fungi remain where they are wanted and nowhere else. Researchers also work on strain selection and engineering, choosing species that grow predictably, produce desired metabolites, and lack traits that might cause problems for crew or equipment. There are questions about long-term genetic stability. Will fungi kept in space for months or years accumulate mutations under radiation and microgravity that change their behavior? Ongoing experiments track these possibilities, comparing space-grown strains to their Earth-bound relatives. And perhaps most importantly, any system that integrates living fungi into critical infrastructure, food, air, or habitat must be designed so that failure modes are understood and manageable. Biological elegance is wonderful, but in deep space, redundancy and reliability are non-negotiable. There is something quietly reassuring in the thought that, even on a distant world, we might rely on the same fundamental partnerships that sustain life here. A habitat on Mars, warmed by sunlight and sheltered beneath a dome, could have mycelium in its walls, mushrooms in its kitchens, and fungal bioreactors humming in its life support systems. Waste streams that would once have been thrown away become food and building material. Radiation that would once have been only a threat becomes, in some tiny fungal cells, just another part of the environment they have learned to endure. You can imagine, if you like, an astronaut in a small, softly lit cabin on a Martian evening. Outside, red dust drifts over the landscape. Inside, a tray of pale mushrooms has just finished fruiting, grown on shredded plant stems and cloth. The air smells faintly earthy. The walls around them are lined with panels that were not forged in a foundry, but grown. Mycelium woven through local regolith and fibers, hardened into a light, strong shell. Behind those walls, in hidden compartments, darker fungi rest as part of a layered shield against radiation. A life support unit quietly cycles water and waste through tanks where fungi and other microbes break, build, and rebalance the chemistry of the little world inside. In that scene, fungi are not alien stowaways. They are crew members, silent and tireless, helping turn a lifeless place into something that can support breath, food, shelter, and maybe even a small patch of green. They bring to space what they have always brought to Earth: the ability to connect things, to transform what has been discarded into something new and useful. Let's come back down to Earth for a moment, but keep that sense of quiet wonder. Because here, under our own feet, mushrooms are already busy with something just as extraordinary. They are cleaning up our mess. If you picture a forest after a storm, fallen branches, rotting leaves, tangled roots, it might look chaotic. But fungi see something different: a feast. Their enzymes break dead matter down and return it to the soil, closing the loop. Scientists realized that if fungi can unravel the complex molecules in wood, they might also be able to tackle many of the stubborn pollutants we've spread through the world: oil, pesticides, dyes, plastics, and heavy metals. Many mushroom-forming fungi, especially the white rot fungi that digest lignin in wood, release powerful oxidative enzymes into their surroundings. These are molecular tools strong enough to crack apart the tough, irregular structure of lignin, one of nature's hardest biopolymers. In contaminated soil or water, those fungal enzymes can attack petroleum hydrocarbons from oil spills, pesticides, and herbicides lingering in fields, industrial dyes staining wastewater, and other complex organic chemicals. The enzymes don't just make the molecules smaller. They often transform them into less toxic compounds, or into forms that other microbes can finish breaking down. This is biodegradation, pollution being slowly translated back into simpler pieces that can re-enter natural cycles. Alongside biodegradation is another, quieter mechanism: biosorption. Fungal cell walls are rich in chitin, glucans, and other molecules with binding sites that attract metal ions and charged pollutants. The surfaces of mycelium and even dried mushroom tissue can act like sponges, grabbing onto heavy metals such as lead, cadmium, copper, and zinc from water or soil. When this happens, the metals are not destroyed. They're immobilized or concentrated. They're less free to move around, less likely to be taken up by plants or animals, and easier to capture and remove. In some systems, the fungal biomass with bound metals can be harvested and processed, turning widespread contamination into something more contained. To really feel what this means, it can help to look at a few specific cases. Imagine a patch of soil soaked in oil, thick, dark, sticky. Traditional cleanup methods might involve scraping the soil away, burning it, or using harsh chemicals. Species like oyster mushrooms have shown a surprising ability to grow on oil-contaminated substrates and degrade components of crude oil and used engine oil. In agricultural landscapes, fungi can help with pesticides and herbicides. Many of these chemicals are designed to resist quick breakdown, so they can protect crops for longer. But that same stability can lead to accumulation in soils and waterways. White rot fungi and other saprotrophic species have been shown to degrade a range of such compounds, often using the same lignin-degrading enzymes, turning them into less harmful derivatives. Then there is the problem that has become almost emblematic of our time: plastic. Thin bags caught in trees, microplastics in rivers and oceans, fibers in the air and soil. Plastics are designed to resist decay, and many do so for decades or centuries. Yet, in recent years, scientists have begun to document fungi that can colonize and degrade synthetic polymers. Some species can adhere to plastic surfaces, form biofilms, and secrete enzymes that slowly cut long polymer chains into shorter fragments. The process is still slow and depends heavily on conditions and plastic type, but it is real. In some experiments, fungi have visibly pitted and thinned plastic films, or reduced the mass of plastic samples over weeks to months. We are still far from a magic mushroom that eats all the plastic, but a door has opened. At the center of all this work is the mycelium itself, the vast, often invisible network that threads through substrates. It is mycelium that gives fungi such enormous surface area for contact with pollutants, and such flexibility in where they grow. A forest floor streaked with oil, a landfill edge, sediment in a drainage ditch. Mycelium can weave through all of it, secreting enzymes wherever it meets something it can use or alter. If you follow that thread a little further into the future, something even stranger starts to appear. Laboratories and design studios where people are not just cleaning with fungi, but building with them, wiring them into sensors, feeding them waste and asking them to grow into packaging, walls, fabrics, even parts of electronic devices. It's as if humanity has finally looked at this old forest partner and said, "You are very good at making things from almost nothing. Would you like to help us do it more gently?" All over the world, researchers are treating mushrooms and mycelium as a kind of living toolkit for sustainable technology, a way to replace plastics, soften our dependence on fossil fuels, and weave biology into places that once belonged only to concrete and wires. One of the most tangible examples is mushroom-based materials. If you take agricultural waste, sawdust, straw, husks, cardboard, and inoculate it with a suitable fungus, the mycelium will begin to grow through it, binding the loose particles into a single solid mass. Once it fills the mold, you can dry or heat the material, stopping growth, but leaving the structure intact. What remains is a mycelium composite, lightweight, strong, insulating, and fully biodegradable. Companies and research groups are turning these composites into packaging, offering them as replacements for expanded polystyrene foam and other petroleum-based plastics. Mycelium packaging can be shaped to cradle electronics, bottles, or food. When its job is done, It can be composted instead of lingering as microplastics for decades. Industrial and academic studies show that such materials can rival or even surpass some conventional foams in mechanical strength while decomposing safely at the end of their life. In Alaska, researchers are testing mycelium-based insulation made from dead spruce trees, looking for breathable, compostable panels that keep homes warm in Arctic winters without relying on fossil plastic foams. When you imagine entire buildings grown from fungal composites instead of poured from concrete, you can feel technology bending closer to the rhythms of biology. In the far future version of this, you might imagine walls that quietly monitor humidity and strain, sending a soft signal when something is wrong, or lightweight panels in remote environments that can patch themselves after minor damage instead of needing replacement. For now, we are in the early, experimental stages, but the idea that you might one day live inside a structure that is partly alive, feeling, and healing itself is no longer just science fiction. In the medical and pharmaceutical world, mushrooms are also pulling us forward. Edible and medicinal fungi produce a wide array of compounds with physiological effects of immune modulation and traditional healing. In agriculture and food technology, fungi are stepping into a starring role. On the farm, mushrooms are being developed as biofertilizers and biocontrol agents, helping crops grow with less synthetic fertilizer and fewer chemical pesticides. At the same time, mycelium itself is becoming food. Through controlled fermentation, certain fungi are grown as mycoprotein, a fibrous, high-protein biomass that can be shaped into cutlets, mince, or fillets as alternatives to meat. Compared with many animal proteins, mycoprotein tends to have a much lower carbon and water footprint, while offering good amino acid profiles, high fiber, and relatively low fat. New studies and startups are exploring how to fine-tune texture, flavor, and nutritional properties, aiming to make fungal protein a mainstream part of global diets, especially in a world that needs to feed more people while using fewer resources. The mushrooms themselves don't know they are part of innovation. They are simply doing what they have always done: binding, sensing, breaking down, building up. But in listening to them, in watching how they solve problems of structure, recycling, and resilience, we are beginning to borrow their strategies for our own tangled century. And now, imagine yourself on a slow, gentle walk. Maybe in a forest you know, or one you invent. The light is soft, as if it's late afternoon or early evening. The air is cool, but not cold. Each step is quiet on leaves and soil. Somewhere nearby, a bird gives a last call before night, and then it's mostly just your breathing and the faint sound of a breeze moving through branches. You pause and let your attention sink down from the tall trees to the understory to the forest floor. And there they are, mushrooms. Sit down on a fallen log, one without mushrooms so you're not squashing anyone, and just feel the support of the wood under you. You know that under the bark, fungi are slowly at work. Beneath the log, mycelium is stretching into the soil, meeting roots and stones and other threads of life. Above you, leaves tremble softly. Around you, the air tastes cool. You don't have to do anything. In this moment, just notice and breathe. Notice how still a mushroom is and how much work is happening around it and through it. If you'd like to carry one thought with you when this story ends, it could be this. Whenever life feels fast and noisy, you can think of mushrooms. You can remember that somewhere, under a forest or a field or even a city park, mycelium is moving slowly and steadily, recycling, connecting, healing. You can remember that strength doesn't always look like speed or volume. Sometimes it looks like soft persistence in the dark. And maybe next time you see a little mushroom after rain, on a lawn, by a path, on an old stump, you'll pause for just a heartbeat. You'll look a little closer. You'll feel, for a moment, the presence of that quiet, hidden network. Then you'll move on with your day, just as the forest does. But a small part of you might be walking a bit more gently, keeping time with the patient, underground rhythm of the fungi that holds so much of our world together, while we live our lives above. And you, right now, rest and drift into the soft, sustaining dark of sleep. Good night.